Chapter 17, Transcription of RNA, Processing and Translation. So this is the nitty-gritty of understanding transcription and translation, both in prokaryotes and eukaryotes. In this chapter, you will learn how information in gene directs the synthesis of RNAs and proteins. And we'll be looking at how DNA is transcribed into RNA and how this is done in both bacteria and eukaryotic cells. Then we'll talk about translation for the second part. That's the second part of central dogma. And we'll take a look at the structure and function of transfer RNA and the structure and function of ribosomes. So this is a very interesting and detailed chapter. This is like going from driving a car to actually looking under the hood and understanding how the motor runs in your car. Let's take a look. Introduction to transcription, RNA processing, and translation. So proteins are the stuff of life, and they serve various roles in the cell. And the reason why proteins are so important is because we're made of proteins. When we talk about a trait or a gene coding for a trait, aren't we really talking about a protein? So a cell builds the proteins it needs from instructions encoded in its genome. And that's why central dogma is so important. So transcription is going from DNA to RNA, and translation is from messenger RNA to protein. And we'll be looking at both of these in great detail, transcription and translation. So RNA polymerases synthesize RNA, and that sort of makes sense. DNA polymerase synthesizes DNA, RNA polymerase synthesizes RNA. And the RNA version of instructions are stored from the copy that it made from DNA. So how is it that actually messenger RNA is being built? It's using activated nucleotides, which are called ribonucleoside triphosphates. And how do we know which one it is? Through complementary base pairing. The one thing that you need to remember, instead of apple tree chewing gum, remember there is no thymine in messenger RNA. There's only uracil, so it's apple umbrella chewing gum. So here we see a replication bubble. There's a non-template strand and a template strand. So the template strand is the one that's being made a copy of. And notice RNA polymerase moves in one direction. It moves in the 5' to 3' direction. And notice that the template strand is 3' to 5. So the opposite of that in an anti-parallel is 5 to 3. So notice that activated nucleosides are being joined to form a copy of messenger RNA. So messenger RNA is going to copy a section or a gene that's found on DNA. And we can see this is exactly how it's done. So I really appreciate figure 17.1. Like DNA polymerases, RNA polymerases have directed synthesis. In other words, they can only move in the 5' to 3' direction. And I think that will be easy to remember. Now, the one thing that you noticed in the other version or the previous figure is that we needed no primer to make a strand of messenger RNA. So no primer is needed. Now, we're going to talk about both bacteria, which are prokaryotes, and then eukaryotes. A eukaryotic cell would be a plant, animal, or fungal cell. So bacteria have only one type of RNA polymerase, and eukaryotes have 3 types, 1, 2, and 3. So we're going to talk about bacteria first, and then we'll go to eukaryotic cells. And I don't know about you, but for me, the first couple of times I read my chapter, I was really confused. I had to tell myself that it's talking about transcription, but first it's going to talk about it for bacteria, and then later on for eukaryotic cells. So try not to confuse the two. Okay, so let's talk about bacteria. So initiation is the first phase of transcription. And when we look at bacteria, they have this protein called the sigma protein. And RNA polymerase plus sigma forms this thing called the holoenzyme. And you will definitely need to know this for your final exam. And it says sigma recognizes sites called promoters. A promoter region is saying, like a store sign or a restaurant that has the open sign and it's blinking, open, open, I'm open. Okay, well, that's a promoter region. And usually that will be found upstream from the gene that is going to be copied. So notice this holoenzyme attaches actually to DNA before any transcription begins. RNA polymerase transcribes genes and is the core enzyme. So what are the promoter regions? The promoter regions are at -10 and at -35. And notice we're using this term or the terms upstream and downstream. So upstream is way before the coding region and downstream is after it. So we'll talk about upstream and downstream when we're talking about a location on DNA. So it says bacterial promoters are 40 to 50 base pairs long. You don't have to memorize any of these sequences. You don't need to know TATAAT or TTGACA. But do you need to know that the promoter regions are at a -10 and 35 boxes? And the answer is yes. And that they're upstream from the +1 site, which is where we began to code or transcribe the gene. This is a great diagram because it shows the holoenzyme. It tells you what part is sigma and which part is RNA polymerase. So notice we have sigma, we have RNA polymerase, which is also called the core enzyme, and it's in green here because remember, aren't all enzymes proteins? And then we have DNA in red. So I love the bottom part picture where it shows you exactly what happens. So notice sigma is the first to make contact with DNA, and it's doing so upstream in the -10 and -35 boxes. So notice you see the two strands of DNA, and you notice that it has that +1 site, and then downstream you'll see -35 and -10 boxes. So sigma connects to the promoter regions, and then afterwards DNA will become in contact with the core enzyme or RNA polymerase. So it says here transcription begins when sigma binds to the -35 and -10 boxes. So sigma is the first part that initiates this copy of going from DNA to messenger RNA. It says sigma can only bind in one direction, and that really shouldn't offend you or bother you because we've been talking about directionality for so long. So the promoter orientation determines which DNA strand will be the template, and it also determines the direction that polymerase will move. RNA polymerase opens the DNA double helix. It creates a bubble, and so instead of calling it a replication bubble, because we're going to DNA to messenger RNA, we will call it a transcription bubble. So RNA polymerase creates this transcription bubble, and so now RNA is going to be actively synthesized. So we have incoming nucleoside triphosphates. Keep in mind they're activated because they have a triphosphate tail, and energy will be needed to form the phosphodiester linkages. And we know the order, and the order is based on complementary base pairing. In this diagram here, we see that sigma is the first to contact bacteria DNA at the promoter region, which is the -35 and -10 boxes upstream from the beginning of the gene, which is at the +1 site. We see that RNA polymerase is in fact filled with a lot of grooves. And these grooves are needed because we need to separate DNA the two strands of DNA apart to create this transcription bubble. And we also need to have NTPs, or activated nucleotides, come in, and we need to have the finished product come out that we see in step 3. So this is how a copy of messenger RNA is synthesized in bacteria. The next phase after initiation is elongation. So when we're talking about transcription, what does elongation mean? Elongation means that RNA polymerase reads the DNA template in the 5 to 3' direction. In other words, nucleotides have to be added to the 3' end of RNA. After elongation is termination. So how do we know that RNA has been completed, the synthesis of RNA for a gene? We know this because it forms this hairpin structure. And this hairpin structure, that means the end sort of folds, almost like it looks like it's making a knot. And this is how we know that termination has happened. And this hairpin structure causes RNA polymerase to separate from the RNA transcript. In this figure 17.4, we can see termination of transcription in bacteria. So here there's the production of messenger RNA, but then it hits the very end and RNA doubles over on itself in step 2, causing this hairpin structure, and this terminates transcription. Now let's move to eukaryotes. So when we talk about eukaryotes, we're talking about linear chromosomes found in plant, animal, and fungal cells. Okay, so instead of having a -10 and a -35 box, actually it says here that there's a Tata box. And this Tata box is where these general transcription factors, instead of sigma, will actually bind to the DNA molecule. Now, keep in mind, another difference is that in eukaryotes, instead of one RNA polymerase, there's actually 3 different types. And then another difference is that instead of forming a hairpin, there's going to be a poly A tail and actually also a 5' cap. And this will let us know that this is the termination of transcription. And the last difference between eukaryotes and bacteria is that transcription occurs in the nucleus and translation occurs in the cytoplasm. Whereas for prokaryotes, they both occur in the cytoplasm because there is no nuclear membrane. If you want to look at this video, please go to the other presentation posted in Blackboard. Okay, so let's now talk about RNA processing in eukaryotes. In other words, we're talking about transcription again, but in a different type of cell. So we've already talked about bacteria, and now we're moving on to eukaryotes. So what it's saying here is, in bacteria, once transcription was done, we had a functional strand of messenger RNA, but that's not the same in eukaryotes. Actually, there's pre-messenger RNA, and then there's mature messenger RNA. So how can we tell the difference between the two, and how do we get from pre-messenger RNA to mature messenger RNA? So in 1977, actually Robertson Sharp found that DNA had coding, but it also had non-coding regions. So not all of DNA is going to code for proteins. There are other regions that contain promoters, and then there's just non-coding regions. And so there's introns and exons. So they found that there were non-coding regions in DNA. And the way that they found this is when they looked at a mature piece of messenger RNA, they found that it was much, much shorter than the original strand of DNA, and that there were loops. These loops were then excluded, and so there were many stretches of DNA not present in the final version or the mature version of messenger RNA, and this is how we know that there are non-coding regions of DNA. Okay, so this describes exactly what's happening. So in red you see a long strand of DNA, and in yellow is a messenger RNA. Notice that the messenger RNA is much shorter than the DNA, and what's happening is that the messenger RNA is excluding many of the loops that are found in DNA. So those are non-coding regions in the loops. So how is it that we get this mature piece of DNA and that we actually splice out the non-coding regions? So primary RNA transcripts contain both exons and introns. Exons are the expressed or coding regions. Introns are the non-coding regions. So how is it that we splice out the introns? So introns are removed by splicing, and there's a group of actually proteins, little molecular scissors called SNRPs. And I'm not making this up, that's actually pronounced SNRPs. And in my mind, to be honest, I think of Smurfs, those little purple or blue cartoon characteristics, because I can visualize them like maintaining or creating a piece of messenger RNA. So I see like little Smurfs, but I'm really thinking about SNRPs. So that's my little mnemonic device. Hopefully you'll come up with one. So anyway, these SNRPs actually help splice out the non-coding regions. In other words, they're going to remove the introns. So this is a 4-step process. So SNRPs bind on to the 5' exon intron and the 3' intron exon boundary. In other words, they're bracketing a non-coding region. And then if they form a spliceosome, and this spliceosome actually connects to form like a little noose or a loop or a lariat, but it forms like it connects together forming a loop. And then the loop is then cut off, and then it degrades on its own. So I love this diagram. This diagram shows everything that we've been talking about until now. So let's look at the very top of 17.6, and we see that DNA has coding and non-coding regions. Coding regions are exons, and they're indicated in dark red. Introns are non-coding regions, and they're indicated by pink areas. So notice we have coding and non-coding regions. When a copy of DNA is made in eukaryotic cells, it has both coding and non-coding regions. But then notice that the mature piece is a solid yellow. So the light yellow from the non-coding regions is actually spliced out. And this is done with the SNRPs down below. So the SNRPs act as molecular scissors. And how they do this is they form a loop and then they excise the loop. So all that's left are exons and all the introns are deleted. Adding caps and tails to transcripts. So when we make our copy, how can we tell that it's completely mature? Well, we can tell that it's mature because it doesn't have any non-coding regions. But another way that we can tell that messenger RNA is mature is that it has a 5' cap and a poly A tail. So there's something at one end and at the other end. And is that important? And you have to remember that in a eukaryotic cell, messenger RNA is formed in the nucleus, but it must exit the nucleus and travel into the cytoplasm for translation to occur. So the 5' cap and the poly A tail are indicative of maturity, but it also protects the strand of messenger RNA. Now, there are still some untranslated regions, but they're only found at the beginning and the end of the messenger RNA. This is another fabulous figure. This shows a piece of mature messenger RNA for eukaryotic cells. Notice on one end we have a 5' cap. We have some methylated bases, and then notice that there's a short piece of untranslated region. Then there's a large coding region, untranslated region again, and then the beginning of the poly A tail. So what does poly A tail mean? It means a lot of A's. So here you have a mature piece of messenger RNA found in eukaryotes. So this is a big difference from that of what we saw for bacteria. So transcription is totally different. Please know the differences. Now let's move on translation. Remember, translation is going from messenger RNA to protein. So in a eukaryotic cell, this happens in an entirely different place. Transcription happens in the nucleus, and translation happens at the ribosome in the cytoplasm. So the sequence of messenger RNA bases is converted into an amino acid sequence, and it takes a lot of steps. So let's take a look at these steps. Now it says here, translation is carried out with the use of ribosomes, messenger RNA, and transfer RNA. So you need to know the players for translation, and there could be a test question asking what is needed to carry out translation. So the answer would be ribosomes, messenger RNA, and transfer RNA. So they did an experiment to show that ribozymes are actually the site of protein synthesis. So an experiment was done, and this showed that ribosomes are very important for translation. When I first took microbiology, and this was as a graduate student because I had conveniently skipped microbiology as an undergraduate, I had a microbiology instructor tell me, if you understand this diagram, you understand transcription and translation. And to be honest, the first time I looked at this, the first 100 times I looked at this, it looked like a falling Christmas tree, because one end is pointy and then the other end, the branches are getting longer and longer. So I'm going to describe why this is happening. So let's see if I can help out. Why is it that these branches are so long over here? Well, this right here, let's start with transcription and translation. So if we look at this, it says here, this is electron micrograph coupled of transcription and translation in bacteria. So they're happening simultaneously. This would never happen in a eukaryotic cell. So let's see here, the trunk of the tree is actually DNA. And these little green things that you see here running through are actually RNA polymerase. Okay, so this one starts here and actually goes all the way to here. And that is why this branch is so long. This one over here actually started at the beginning. RNA polymerase started at the beginning, and it only ends up here. And that's why this branch is of medium-sized length. And then this RNA polymerase started at the beginning, and it's only gone a short distance. And that's why this messenger RNA is very short. So remember, all of the RNA polymerases started at the very, very beginning. The other thing to note is that we have ribosomes, and because bacterial cells don't have a nuclear membrane, everything's out in the cytoplasm, the ribosomes are right there. So the question is why is this protein so long? Why is this one a middle length, and why is this one so short? Remember, ribosomes read messenger RNA from the 5' to 3' direction. So this ribosome number 1 started all the way here at the very beginning. It started here and it's finished reading all the way down here. And that's why this tail is so long. Ribosome number 2 also started at the tip and it's only traveled midway, and that's why this protein strand is of medium length. And ribosome number 3 just started, and that's why this protein is very short. The same thing goes for the protein formation here. Now, when I want to make 10 copies or 20 copies of something to give as handouts to you as the class, I go to the copy center and I just punch 20 on the copy machine. But in this case, if we're going to make a lot of copies of a gene, we're going to need a lot of RNA polymerase and a lot of ribosomes. So let's see how many copies we have here. We have 1, 2, 3, 4, 5, 6 copies of the same part of a DNA strand of a gene. So this is how a bacterial cell is going to make multiple copies of a gene. So in bacteria, ribosomes begin translation on messenger RNA before transcription is complete. In other words, they're happening at the same time. It's simultaneously. And this is because there is no nuclear membrane. So we can have multiple ribosomes attached to messenger RNA, just like we saw in the previous diagram. And this is called a polyribozyme. This is like making many copies, or me going to the copy center and asking for 7 copies. Okay, so many copies are being made at one time. In eukaryotes, transcription and translation are separated. And the reason why they're separated is because of the nuclear membrane. Transcription happens in the nucleus and translation happens in the cytoplasm. And you know for sure that this comparison will be asked of you on the exam. Okay, so how does messenger RNA specify the amino acids? Okay, so before we get started on this, I just want to step back and tell you a little story. Most of you know that I have 3 children, a daughter and 2 sons. And I remember when they were teenagers and getting ready to get their driver's license, I was excited and scared at the same time. And I was excited because I didn't have to take them places anymore. They could drive themselves. And then if I actually needed help, they could help me out too. But I was scared because, well, they could have a car accident and they're becoming more independent. So with their independence, with wheels means freedom, right? So you can go to places without actually being chaperoned all the time. So I was sort of scary excited. And that's sort of how I feel about this part here. Is how does a messenger RNA specify the amino acids? Well, now we're going to be introduced to a third type of RNA. And you say third type, didn't we just talk about one type, messenger RNA? And the truth is, yes, we've only talked about messenger RNA, but the truth is that ribosomes themselves are a type of RNA, and we call that rRNA. So that means ribosomal RNA, and the third type of RNA is transfer RNA. So transfer RNA is going to be important for translation. Okay, so here they came up with this hypothesis. Is it messenger RNA and voila, all of a sudden you have amino acids and then you have the formation of a protein, or is something else happening? What is happening? So here was the ideas back in the day, like when I was born in the 1950s, is from the messenger RNA do we get just amino acids that understand the triplet code? Or is there some molecule in the middle that you see in hypothesis 2 called the adapter molecule? Well, the answer is there's some molecule in the middle called the adapter molecule. And those black triangles represent transfer RNA, the third type of RNA. So they did this experiment, and what they had done is they had messenger RNA and ribosomes in a test tube, and by serendipity they all saw that over a few minutes, like none of the amino acids were there anymore. Where did they go? and they had been converted into a protein, and it's because there was the secret ingredient and that you see in black here, the transfer RNA, which looks like an upside down L. And at the end, it's carrying leucine as its cargo. So then they realized just by mistake, because of this experiment, that there was a magic or mystery molecule, and this mystery molecule was transfer RNA. So it is the adapter molecule used in translation, and it is called transfer RNA. So an amino acyl transfer RNA is a transfer RNA that is linked to an amino acid. In other words, it's carrying cargo. When it's not carrying cargo, it's just transfer RNA. If it has cargo or an amino acid attached to it, it's called amino acyl transfer RNA. So what does a transfer RNA look like? As I mentioned before, it's made out of RNA and it has loops. It's RNA folding on itself so that it looks like an upside down L. And at one end, they have this sequence here called CCA. And this CCA is found at the 3' end. And this is where the cargo, the amino acid, binds to transfer RNA. At the opposite end of the upside down L, there's a loop, and it contains the anticodon. And this anticodon matches to the codon found on messenger RNA. And this is how the transfer RNA knows to land in a specific place, because it has the anticodon that complements the codon. In this drawing here, we see that the mystery ingredient transfer RNA is in black. And we know that it's the third type of RNA. And notice we see a codon on messenger RNA and an anticodon on transfer RNA. The anticodon is UGA. And notice it complements ACU. Now, at the opposite end of transfer RNA, on the CCA side, it is loaded with an amino acid called serine. So this is an amino acyl transfer RNA. ATP is required to actually load the amino acid onto or to bind the transfer RNA. So what is loading the amino acid? It's an enzyme, and this enzyme is called amino acyl transfer RNA synthetase. In other words, it's charged and it's able to catalyze the reaction. Now, it says there are 20 amino acids, and each has a different amino acyl transfer RNA synthetase. So there's a special one for each of the amino acids. In this diagram here, we can see how amino acyl transfer RNA synthetase actually loads or binds the amino acid onto transfer RNA. So notice at the CC end, you see the amino acid, and the big green glob with a lot of molecules is amino acyl transfer RNA synthetase. It's like one of those great big husky moving guys that's actually loading the cargo, which in this case is the amino acid, onto transfer RNA. Now it says here there are 61 different codons. Now let's stop and let's think back to why we have 61. Because remember, there's a total of 64 combination, 4 times 4 times 4 in the triplet code. But 3 of them are stop codons that just say stop. And there's one start codon, AUG, but AUG codes for methionine. So 60 plus 1, which is the AUG, makes 61. So there's 61 different codons, but only 40 transfer RNAs. And it goes to this idea of the wobble hypothesis. That means for most of the time, the first two bases in the triplet code are important, but the last one sometimes is important, not all the time. And we call this the wobble hypothesis or wobble pairing. So most of the time we're looking for the first two, And the third one sometimes plays an important part, but most of the time it's the first two. So we don't need that many transfer RNAs. So 40 is enough to actually transfer the 20 different amino acids. Ribosomes contain many proteins. And so this is ribosomal RNA, as I had mentioned earlier. This is the second type of RNA. So we have messenger RNA, rRNA, and tRNA. So this ribal RNA is what actually makes the ribosome itself. Now let's focus on the ribosome because remember we need it for translation. Well, it turns out that the ribosome has two parts. It has a small subunit and a large subunit. You know what it reminds me of? It reminds me of a hamburger bun. You know, the fancy ones with either dried onion on the top or sesame seeds, where the bottom part of the bun is small and the top part is large. So then we have a large and small subunits. So it says here during translation, 3 transfer RNAs line up within the ribosome. So what it means is the ribosome itself has spaces for 3 transfer RNAs. And these positions are called APE, A, P, and E. The A site is to accept a transfer RNA. The P site is to form a peptide bond, a peptide linkage between one amino acid and another. And the third position is to exit. Now remember, before we go on any further and we think of APE, the 3 sites within a small little tiny ribosome that has a small and large subunit, isn't it true that this little ribosome is a mighty force? Because it is reading the messenger RNA. So this ribosome is always in action. The first site accepts, the P site forms a bond, and then the E site exits the unloaded transfer RNA. This is a fabulous diagram of a ribosome. Here you can see the small subunit and the large subunit. And you can see the 3 sites, APE, A, P, and E. And notice as it's going from left to right in the 5' to 3' direction, notice that it's moving along messenger RNA. And notice that the A site, which is the first site, it's accepting new transfer RNAs. In fact, the proper name is amino acyl transfer RNAs because they're loaded with an amino acid. Then it moves over one other site because it's keeping on moving on to the 3' direction. Then the transfer RNA moves to the P site, and there it's going to bind with other amino acids that have been left. And then the ribosome will move over one position, and then it'll go to the E site. And notice at the E site, it's unloaded. It's an empty transfer RNA, so it's now going to exit. So these are the 3 sites that are found in a ribosome. So it says ribosomes synthesize proteins in a 3-step sequence. And those 3 steps match the A, the P, and the E sites, APE. A is for accepting, P is for forming the peptide bond, and E is for exiting. The empty transfer RNA that does not have a amino acid bound to it will now be released and go out to the cytoplasm and find another amino acid. So just like transcription, there are 3 stages: initiation, elongation, and termination. So we had initiation, elongation, and termination for transcription, and we'll have the same 3 phases for translation. The protein grows by 1 amino acid with each repeat of the 3 steps. So here we go. What is happening in initiation? So initiation phase of translation begins near the AUG start codon, but it actually happens upstream. So upstream or before the AUG start codon is the Shine-Delgarno sequence, or the ribosome binding site. So this is where actually the the ribosome and the messenger RNA bind to each other, and this is about 6 bases upstream from the start codon. And this is done with the help of initiation factors. The first transfer RNA is called the initiator, but you guys know what it is. It's methionine because AUG is always the first start codon. Translation initiation is a 3-step process. The first step is that messenger RNA binds to the small ribosomal subunit. Then the initiator transfer RNA, the one that contains AUG or methionine, binds to the start codon. And then the large ribosomal unit binds so that the initiator transfer RNA is in the P site. Then translation is ready to begin. You know, that's a mouthful. So what does that mean? That means exactly this diagram. It's sort of like when you have, going back to the hamburger buns for the ribosome, it's like you build your hamburger and then you put on the top at the end. So you put on mayonnaise and lettuce, your hamburger patty, and when it's all loaded, then you put on your hamburger bun. And that is exactly what's happening here. Now, to align all of the ingredients on the small subunit, remember upstream around 6 bases, we have the Shine-Delgarno sequence. So it is upstream or before the start codon AUG. Now notice AUG fits into the P site. It's not the A site, but the P site in the middle. So notice here, that is the first one. And because we're talking about bacteria here, it's called FMET. So there's additional molecules associated with methionine, which we will not get into in detail, but we see that there's that little F, and that to me indicates that we're talking about bacteria. So we have our Shine-Delgarno sequence. The start codon is in the P site, and now we put on the large subunit onto the ribosome. So now we're ready for translation. So this part is considered initiation. Okay, so the next phase is elongation. So what's happening in elongation? The ribosome starts to read messenger RNA in the 5 to 3' direction. As it's reading, remember, transfer RNAs will land on the messenger RNA because of the anticodons. Remember, anticodons complement the codons. So it says at the start of elongation, the initiation transfer RNAs in the P site. The E and A sites are empty. But as it starts to move, then the A site will accept a new transfer RNA. So amino acids at the P and A sites will then become activated. So before we go on any further, it says, is a ribosome an enzyme or a ribozyme? And the answer is it's going to be a ribozyme because remember we had talked about an RNA world first. So RNAs are very like magical molecules. You know, they can be part information molecule, nucleic acid, and they act like a protein. So it's like RoboCop, right? So they can catalyze reactions. So a ribosome is in fact a specific type of ribozyme. And so as it moves along messenger RNA, it's here are the 3 sites, A, P, and E, A, P, and E, A, P, and E, A, P, and E. It's moving over one site at a time, one site at a time. So it just moves one site at a time. And when I say site, I'm talking about the A, P, and E sites. And so it says here elongation factors help move the ribosome. So this movement along the messenger RNA in the 5 to 3' direction is called translocation, and it's moving one site at a time. So if you look in your book, it'll be across over 2 pages in your book, this movement, this translocation. So the ribosome, as it slowly moves, you can see that the transfer RNAs start moving to different sites. So notice in step 1, at the A site for accepting, it is accepting a new transfer RNA. Notice the anticodon at the bottom, and it's carrying cargo at the top at the CCA end. And then notice in step 2 that a bond is going to form, and notice now the ribosome is going to move over one. So now that red colored transfer RNA has moved to the P site. And when the ribosome moves again, it's that red colored transfer RNA is going to move to the E site. Okay, so moving down the messenger RNA. So it arrives, a peptide bond is formed, and then the transfer RNA exit, and the ribosome moves. So this is going to continuously happen as the ribosome moves down the messenger RNA to read it and to form a protein. So here's the end of that. So notice that the red transfer RNA is now eventually moved to the E site where it will exit. And so a new transfer RNA will go into the A site, and this one is indicated by blue. And this one transfer RNA coming in and the other one exiting, coming in and exiting as the ribosome moves down the messenger RNA. Now, how do we stop translation? What is the termination of translation? Well, there's this thing called a release factor. What is a release factor? It's sort of like a plug, and it enters into the A site. So it looks like a transfer RNA in size and shape, but what it's actually doing is hydrolyzing or breaking the bond, linking the P site to the transfer RNA. And when it does this plug actually stops translation. So it says here the newly synthesized polypeptide or protein, the transfer RNAs and ribosomes all separate and disengage from each other. So let's take a look. So all of a sudden we see a release factor which is purple. It has the same shape as transfer RNA, but it's different. And what it's going to do, it's sort of going to clip off everything, and when it does, the plug remains there, and it's sort of like a clipping mechanism which releases everything, and everything is released and disconnected. And the protein that was read from the messenger RNA is released. The large subunit is disengaged from the small subunit of the ribosome, and the messenger RNA is also released so that other ribosomes can make copies. This summary table is so important because this summary table compares transcription and translation, the differences between bacteria and eukaryotes. And I must admit, the first 2, okay, even 3 times that you read this chapter, it might be confusing. So this is a wonderful guide to show you that transcription and translation are different among bacteria and eukaryotic cells. Please look at this video. It will be in another presentation posted in your Blackboard. After the protein is made, which is the primary structure, a string of amino acids, there's still modification to go. Remember at the very beginning of the semester when we talked about proteins, there's the primary structure, secondary structure, tertiary and quaternary structures. So we have just completed the primary structure, but there's still much more to go. So most proteins go through an extensive series of processing before they become functional. So we often call this post-transitional or translational modification. And these steps take place in various locations of the cell. So we can have these molecular chaperones, and oftentimes these molecular chaperones are actually found in the endoplasmic reticulum. Remember, there's smooth endoplasmic reticulum and rough, and remember the rough ones were associated with ribosomes. So after the protein is made, they usually end up in actually the lumen of the endoplasmic reticulum, and there we can find molecular chaperones. And these molecular chaperones guide and speed up protein folding. Because remember, for a protein to be functional, shape is critically important. And then also sugar and lipid groups may also be added to proteins. So all of this are modifications to after translation. This is a wonderful figure showing all of the steps of transcription and translation in a eukaryotic cell. And how do I know that it's a eukaryotic cell? Because I see a nuclear membrane. And I also see mature messenger RNA, which you would only find in eukaryotic cells. Okay, so notice here it has all of the steps, and we just finished translation, and then notice step 4, post-translational modification. So that is what we've just finished discussing. This is a wonderful figure to study, and if you understand this figure, that means you understand the basic concepts of this chapter. Thank you. We've reached the end of this chapter.